So we will almost start, and the next talk is by Alex Lan, and it's a perfect follow-up of previous talk by combining the pathways and protein interaction networks. And 1 minute to start. Yes.
Hello, I will present a new approach to identify human signaling pathways using context. This is my master's thesis under the guidance of Professor Michal Ziv-Yukelson and Dr. Esti Agirlote. A major challenge in systems biology is to identify the signaling pathways that give rise to specific phenotypes and behaviors. Current techniques often rely on a network representation in which specific nodes represent proteins or genes and edges their physical interactions, and are often associated with a score. However, this network fails to capture the tendency of proteins to favor different partners under distinct cellular conditions. For example, let us look at GRB2, growth receptor binding protein 2. It has many different partners, but only some are relevant in a specific condition. The world without context is gray and boring, preventing us from understanding the condition-specific behavior of GRB2. Now the world is not so boring. We added color using the gene ontology, and basically this represents our context. We can see that GRB2 has many different partners under distinct cellular conditions. For example, under growth factor signaling pathway, GRB2 interacts with these partners, and under DNA replication and repair, with these partners. Our goal is to introduce context into the network and use it to identify signaling pathways. This was done previously by others. In 2006, Scott et al. Used protein localization data from the gene ontology to identify signaling pathways that move from the membrane to the nucleus. In 2007 and 2008, Pandey et al. And Banks et al. Used the protein-protein interaction network to uncover underlying context principles within the gene ontology. The novelty of our work is in the consideration of underlying context principles to uncover novel signaling pathways, and in the consideration of multiple contexts to select a single specific one. Our proposed framework consists of an interactive model, a context-based scoring scheme, and a path interpretation and scoring algorithm. I will now present each one of these parts. Our interactive model consists of nodes that represent proteins and genes and edges that represent their physical interactions. We constructed our network by combining data about experimentally validated physical interactions from several databases, which resulted in over 170,000 interactions that link over 14,000 proteins and genes. Next, we added context to our network using the gene ontology as follows. Each Go annotation was considered as a distinct label, and each node was assigned a label vector of its Go annotations. The goal of our work is to find signaling pathways that link a source protein to a target gene. First, our algorithm finds all simple paths of length 2 to k that link S to T, where k is a predefined user parameter. We used 5 edges. Next, our algorithm scores all of these paths based on the optimal context transition scores. But for this, we first need to define context transition scores. We accomplish this by defining the context transition scoring matrix M, which assigns for each 2 labels a score. How do we compute M? For this purpose, we exploited the Spike database of manually created pathways. We took 21 process-specific maps from Spike and from each extracted all paths that link a protein to a gene with the last edge being a transcriptional regulation edge. We then took these 18,000 and plus pathways and combined them into a set of over 6,000 unique directed interactions. We then used these to compute the conditional probability of observing label Lj after label Li on some path. This score is further fine-tuned to deal with promiscuous labels and common ones, and further details can be found in the paper. Finally, our algorithm ranks and labels each node in each path based on the context transition algorithm. Our algorithm considers all paths that link S to T. How do we score and label a specific candidate path? Let us look at one. Here's a candidate path P. We construct for it the context label graph G as follows. For each node in P is associated with several Go annotations. Therefore, for each node in P, we construct a column of nodes in G. For example, node U is associated with its two contextual labels in G. Next, we connect each node in G by an edge to all nodes in the next column. We assign weight to these edges using our context transition scoring matrix M. For example, the edge L3 to L4. We also add special skip edges that are two columns apart to deal with missing annotations. The weights of these edges is set according to M with an added penalty to discourage their overuse. You can find the details of the penalty in the paper. We can now see that a potential contextual interpretation of P corresponds to some path in G, with the best contextual interpretation corresponding to the heaviest path in G, shown here in red. We compute it using dynamic programming, and the recursion can be found in the paper. Finally, our algorithm reports a single chosen label for each node in P. Note that node V doesn't have the light green label L3 in its column, though it was assigned. This is because it fits best within the contextual interpretation of the rest of the path, and was because of the skip edge over here. I have presented our framework and algorithm. I will now present our application of this framework to show that known signaling pathways are context sensitive, and for the prediction of influenza-related infection pathways. Our first step was to validate whether known signaling pathways are indeed context sensitive. For this purpose, we exploited the spike database mentioned earlier. We used 21 spike maps in a leave-to-out statistical significance test. Specifically, we computed M based on paths included in 19 spike maps, and for the two left-out maps, computed all paths that link a protein to a gene. We then applied our context labeling algorithm for each path in the two left-out maps. Our goal is to check whether our context labeling score is actually statistically significant. We did this by taking all paths in the two left-out maps and all labels for each node in each path. We next shuffled the labels of the nodes, recalculated the path score, and repeated the process. One would expect that a successful path, namely, if known signaling pathways are context sensitive, one would expect that their score based on original labels would be better than their score based on shuffled labels most of the times. So here we have a definition of a successful path. Here we see a result of this analysis. The x-axis represents the path length in edges, and the y-axis the percentage of paths for this length that were successful. We can see that paths of length 3 to 5, most of them were successful. Next, we applied our framework for the prediction of influenza infection-related pathways. During infection, viral proteins interact with human ones to recruit the cellular machinery for viral proliferation. However, The exact pathways and intermediate proteins involved in the infection process are just beginning to emerge. In 2009, Shapira et al. Reported a large experiment in which they identified protein-protein interactions between 10 influenza proteins and 87 human ones, and performed extensive mRNA profiling. They chose over 1,700 candidate genes to check their effect on viral infection by RNA silencing. They found that 616 of them were siRNA positive, namely had a significant effect on viral proliferation or interferon production. Here we took an advantage of this wealth of information to assess our context-sensitive framework. We did this by calculating the set of all simple paths that link the viral interactors to the differentially expressed genes in our network. This left us with 36 proteins and 527 differentially expressed genes that are present in our network, are connected, and have annotations in our scoring matrix M. We then applied our algorithm for these pathways. Here we see the result of this analysis for the top 5% scoring paths. We now see x-axis path length, the percentage of background paths that were successful, namely contain at least one siRNA positive intermediate protein. Here we see 60%, 70%, and 75% for background paths. Now we see the same data for paths that were scored best by our algorithm. We can see that the results are far better than the background. Let us consider all paths of length 4. There were over 3 million paths of this length linking our source and target proteins. But only 49,000 of them were actually scored. This is due to missing annotations and our limitation of only allowing one skip edge. We now see a correlation between the context score percentile and the percentage of paths in that percentile that are successful. We can see that as the score is better, the chance of finding a successful path is much higher. This correlation is statistically significant. Here we see a top-ranking path linking the viral interacting protein TRAF2 to the differentially expressed gene IRF7. This path ranked best out of over 900 paths. And proteins in this path have labels closely related to viral infection. Also, we can note that 2 of the intermediate proteins are siRNA positive. Our proposed framework and algorithm are available as an open web tool called ContextNet at this address. Here you can enter a list of source proteins and a list of target genes. A newer version that will be released soon will also allow you just to enter your own path. Here we see the output. All pairs of source and target ranked from left to right according to their context score. The path, the single chosen label for that node. If we click on a node, we can see all potential other labels that the algorithm could have chosen and the interest gene description of that gene. I would like to thank my wonderful advisors, Dr. Esti Agirlotem and Professor Michal Ziv-Yukelson, the Frankel Center at the Computer Science Department in Ben-Gurion University, the Computer Science Department at Ben-Gurion University, and the ISCB for providing a generous travel fellowship. Thank you. Are there any questions?
Hi.
Yes.
Your method, in a way, is biased to find the frames of each protein in the way because you are finding like the always related to it's a context. All the approach is based in a context.
Yes.
Friendship. So if I call it in general friendship, it's like if a protein is annotated to different this list of go terms or pathways, then the next protein that will go in your path will be something similar, like
not necessarily similar. What we actually
Well, not similar, similar in the sense of the annotated to similar term.
Well, we actually found that it's not a similarity measure, because if this is true, one would expect that the same label would be most similar to itself in our matrix. But we actually found that often the same label is not very similar to itself in our matrix, because it doesn't often appear after itself in a pathway. Pathways encode information. Labels transition from different contexts. For example, we reach a hub protein, it has to make a decision how to proceed, and based on the input context, it switches to another context. And this is what we learn from existing pathways, and we try to encode this information in our scoring matrix. Yes?
So there's already a lot of omics data, such as proteomics and the expression data. Just given that at the time 0 we got proteomics data, and then the time 1 and time 2 is the time series. So from the time 0 of the proteomics, and then we got the T1 expression and the T2 expression. So from T0 to T1 to T2, so is it consistent for your prediction?
We didn't use proteomics, time series proteomics data, so I'm not sure exactly what you meant by your question. We can use time series proteomics data as context and to learn from it for our transition scoring matrix, but this network was built based on physical interactions, protein-protein interaction databases, so there's no proteomics time series data there. Or was that okay? Did that answer your question? Because it wasn't exactly clear.
Oh, we discuss later.
Okay.
Any other questions? We'll have an earlier break. Thank you again.
Thank you very much.